This lecture will cover the Dematiaceous molds. Dematiaceous fungi are characterized by their dark coloration, and this is due to their ability to produce melanin. They are agents of superficial and subcutaneous mycosis. They affect the skin and subcutaneous tissue, and they are ubiquitous to the environment. Humans and animals tend to be accidental hosts. And they are also classified into slow growers and rapid growers. So the slow growers usually mature within about 7 to 10 days, whereas the rapid growers mature in less than 7 days. Sometimes it only takes a couple of days. So here we have a list of the ones that are slow growers versus the ones that are rapid growers. The superficial mycosis caused by these fungi include Tinea nigra, which is a skin infection, and Black Piedra, which is a hair and scalp infection. The two other superficial mycosis we've already covered, which is the Tinea versicolor and the White Piedra. And again, these organisms affect that skin and hair and nails. So they are also diagnosed through the appearance of the lesion or how the infection looks, but also by skin scrapings and looking at the hair shaft. Hordeae wernickeyi is the cause of Tinea nigra. So it causes these brownish, black looking flat lesions on the keratinized skin layers of the hands and feet. And it's common to Central and North America, Southeast Asia, Africa, and Europe. And so these infections are usually treated with some sort of topical antifungal. It is diagnosed only to rule out the possibility of melanoma, because you can see that these lesions do look sort of suspicious as far as some sort of melanoma type spot. And they are typically reviewed or looked at with KOH, and the tissue fragments or the skin scrapings are looked at for any sort of hyphae fragments or any sort of budding yeast. The macroscopic appearance of H. wernickeyi growing on an agar plate will appear as can start off as a yeast-like look to it, and it can usually be dark, as seen up in this top image, or it can be a little bit more of an olive color, and it will start off sort of moist and shiny, yeast-like, but then with age it can start to have more of a mold type look to it. So you can see this colony here, where around these periphery edges it has started to get a bit of a mold-like look. For the microscopic appearance, septate hyphae, so you can see this top image. It's a little hard to see because it is a darker colored mold, so you can't see those septates too good, but if you look at this bottom image, you can see some of those divisions. And it does tend to have a yeast-like look, and with these images you can see how these cells look almost like a little yeast sorta, but these are your annelids, and you can see this bottom image where one of them is coming off and it's starting to bud off there. Piedra hordeae causes Black Piedra, and this affects the hair, which forms a black stony type nodule that cements itself to the hair shaft. And these can be seen with the naked eye, or it can be seen microscopically. So in the top you see a microscopic image where it has formed on the outside of the hair shaft, and at the bottom you can see those dark filaments or those dark deposits on those hair shafts. These can be found in, this infection can be found in Central and South America, Southern Asia, and Africa. And it's been thought to be acquired through swimming in rivers and stagnant waters. And it is treated by shaving the affected area. It can be examined through a KOH prep, and this is done by placing that hair shaft with that nodule on it in between two slides and crushing it with that KOH present. And that will usually reveal the oval asci within that microscopic view. And within this, you will see 2 to 8 aseptate ascospores within that. This organism is a slow grower, so on a agar plate it will appear as a small folded dark brown to black colony. So you can see here at the bottom where it is a smaller colony. It doesn't cover the whole plate. And so you can see how you have the organism growing here. But then when you look out here in the agar itself, you can see where some of the reddish brown pigment has diffused into that agar. For the microscopic appearance, it does not penetrate the hair shaft, so you can see a larger image of it formed on the hair shaft, and then you can see it zoomed in where it does not actually penetrate the hair shaft, but rather forms on the outside edge. And this organism is a very septate organism. So if you look at these hyphae up here in this top image, you can see all of those divisions very easily with all those septa forming within those hyphae. Subcutaneous infections are divided into 4 categories. 3 of those are caused by these organisms, and the 4th, the sporotrichosis, we've already covered with our dimorphic lecture, so we won't touch on that again. But these infections cause lesions that usually start from some sort of trauma that inoculates the site. They're common in the tropics and subtropics. And with these organisms, they do tend to form some sort of granulomatous type infection. And so by looking at the granules that this organism forms, which contains the hyphae of the organism itself, it can help to narrow down what type of organism is causing the infection. Mycetoma infections involve a chronic granulomatous type infection with the lower extremities like the feet, but it also can happen in any part of the body. It causes swelling and a purplish discoloration with a tumor-like deformity of the subcutaneous tissue. And it can invade multiple signage tracts that will drain the pus-type material, which can contain yellow, white, red, or black granules. So the granules that actually come from this infection will have a color to them. It can progress to the bone, muscle, and other tissue that are surrounding that site, and it can lead to amputation. So with the exudates that come from different types of infection, and so this would be like the pus, it can be bacterial or it can be fungal. So we're going to look at the fungal ones. Now with the fungal ones, they can have white grain mycetomas or it can be black grain mycetomas. And so with the white grain and the black grain, it has to do with the granules. It is ID'd by direct examination with KOH or through looking at the granules and observing the hyphae. For the mycetomas, we are going to look at the ones that cause the white grain and the black grain. So the white grain are caused by unpigmented fungi, and of course 2 of these we've covered in the opportunistic lecture. And with the black grain, they tend to be pigmented type fungi. And so if you look in this image here, you can see some of those what they call black grains, and you can see where they just look like some sort of little hard nodule of some sort. So you can see several of those here all around whatever this is, this tissue that's been biopsied. Pseudala Sharia boydii causes mastoma and pheohypomucosis. So with this organism, you do see the white granules, soft white granules, and it can cause a variety of infections from a pulmonary fungus ball to infections of the eyes, endocarditis, or some sort of disseminated infection where it can invade the joints and the brain, things like that. And with these, with the subcutaneous infection, you're looking for the granules, and with the more disseminated infections, you're looking for the hyaline hyphae. Your specimens could be respiratory secretions, gastric washings, skin, the cornea, and you can see an infection here of the cornea where that outside edge is infected, and it looks like maybe some type of surgery or something has taken place on that cornea. There are two reproductive forms. You have an asexual form and a sexual form, and of course the asexual form has an alternative name for that form. This organism is a rapid grower, and for the microscopic appearance, it produces hyaline septate hyphae. So you can see all those hyaline hyphae. The septa in it are a little bit difficult to visualize on this picture, but the conidia can come straight up from that hyphae, or it can come up and form in little clusters. So you can see several of the little clusters of those conidia. For the sexual form, you get this big spore with the sac-like spore with the little spores that are inside of it. So both of these have popped open for those spores to come out. Madurella species cause the black grain infection. And so with the subcutaneous tissue, you can see the hyphae fragments within that tissue. Here you can see an example of those black grains within this tissue sample here. With the macroscopic appearance on agar, it is a slow grower, and the colonies are folded with those little radial folds around the edges. And it can be a variety of colors. With the microscopic appearance, it is dark, thick hyphae, and on those hyphae you have phyllodes that are long and tapering, so you can see some of those where they kind of taper off at the end, and then you get these little flower-like structures here at the end. These organisms are temperature tolerant, so sometimes the biochemical test can help to differentiate one organism from the other, one species from the other. Exophalia genselme has soft black granules in this infection, and within that cyst that affects the subcutaneous tissue, you can have hyphae fragments and budding yeast. It is a slow grower. It starts off with a yeast-like, dark-colored type colony, and then as it gets older, it can have a velvety type appearance. So you can see an example up top where it's starting to get that velvety type appearance to it. And it does use potassium nitrate to grow at 37 degrees. It only grows at 37 degrees. With your microscopic appearance, when the colony is young, it can have a yeast-like look, but as it ages, it gets these stick-like conidia, and so you can see how it comes up, these conidia come up, and then it has these, all of these clustered type conidia at the end, so it kind of has this like little flower looking arrangement as well. So a lot of these have this little flower looking clumps of conidia at the end. Curvularia infections cause black granules. You can have a mycetoma or a pheohypomycosis type infection. It can affect the pulmonary or disseminated type infections, affect the eyes, the brain, endocarditis, or a fungus ball in the sinuses. Specimens could be respiratory secretions, the cornea, the brain, skin, nasal sinus type washes or samples, culture samples. The colonies can appear fluffy with an olive gray type appearance. You can see an example up top where it kind of has that olive color grayish tint to it. The microscopic appearance is septate hyphae, and it can be a kind of a color because these are, melon type pigment, so you can see this image here where it's colored. But you can also see little crooks in it where it kind of bends where those conidia start coming out. So you can see how it kind of angles up with that little notch, and then you can see some little bent areas in it. And those conidia that are attached can be a little grouped as well, or they can be single. But they also will have a curved shape to those conidia, and you can see the arrows here at the bottom where they kind of bulge out in the center. So the ones here in this middle image are a little bit harder to see because they are grouped together, but here at the bottom you can see several of those where they have that bulge in the center.